So I will give a couple of minutes for people to join, and then we'll start, and then we could chat more. I mean, right now. So how Tufts and LA, right now in terms of weather, and LA should be great. That's I kind of love it. But Tufts,
we just started classes last Thursday, so
last Thursday,
our outdoor population suddenly jumped by, I think, more than a factor of 100. Everyone between their classes waits outside rather than being in a building. So it's kind of shocking to see what was an almost deserted courtyard completely, filled with people working on their laptops and having coffee together. And yeah, it's
that's amazing. So you completely, in-person class?
We're hybrid. All our classes have to be hybrid this quarter. So I have about 40 people in my class, and I had 8 online and the rest in person, and separated out in a room with everybody in a mask and so forth.
Wow, that's great. Wow. So yeah, we are also completely in person class right now, unless some people kind of become sick, and then that way they kind of join, kind of online. But we basically everything basically the in-person classroom right now. So our university invests a lot of money to install the filter.
Oh, great.
Yeah, and then even very nice the recording device, every single
oh, really?
classroom. Yeah, so a lot of money actually invested, and then I'm really grateful because the university basically care about the safety and then the education of the student. So how about Tufts? I mean, you are postdoc, probably not that much interested in the classroom right now, I guess, but
so definitely, like Professor Weiss said, the crowd has increased from nothing to actually stormed across the hallways and everything. It's good to see people again in some sense. It's nice in a way. The weather is moving towards fall, so it's getting now colder. So that's again a good change.
yeah, but when I was in Boston this summer, a little bit hotter than I expected. I mean, I would say, global warming or climate crisis. I kind of feel, because at that time it's almost 95 something in Boston in summer. Generally okay without air conditioner, but at that time was wow. And then I think this is the most beautiful time in Boston, right?
Yes.
And LA always beautiful, in terms of weather, except some rainy season, winter, right?
We do have we don't get very much rain where we are. It's really a desert, but the hottest months here are September and October.
Oh yeah.
I'm not entirely certain the offset. We do get the leaves change color because it's really based on the time of day rather than the temperature.
I see.
Our cats drop, shed fur.
So interesting, we now have the both coast and then middle.
Yeah, even in the middle, right?
Yeah, so we are basically the entire nation. Okay, so I guess I will just start because everyone kind of busy. So let's see. So good morning, afternoon, evening. Hola, hello, bonjour, ni hao, konnichiwa, 안녕하세요. And hello everyone. Welcome and thank you for joining us the 6th seminar and ACS Nano Day with the editor-in-chief and ACS Nano. Now we can see the depth of the topics of this seminar series, from DNA storage device, wearable sensors, control theory, recorder, metabolic engineering, and today potentially nanobio and some medical applications. Regarding the diversity, This seminar series has hosted until last week 5 female and 5 male pioneers and 2 female and 3 male rising stars. Nationality were US, Korea, China, India, and Australia. No Europe so far, right? In fact, we have many in the next few months and also next year, and also 5 pioneers from Europe recently joined us for the 2023 season. That means I'm accepting more nomination on a rolling basis, and please send your self-nomination or nomination of your student and postdoc. The eligibility rule is very simple. Untenured professors or people interested in academic job that related to synthetic biology broadly defined. I also saw kindness from this SynBYSS community. As I told you during the 3rd seminar, one of young speakers informed me of great news, tenured. He's willing to give his speaking spot to others. And this week, actually another, Kyle, did the same thing. He recently decided to change his career direction a little bit, and he's pursuing a startup career for a while before coming back to academia. And he wants to give a talk at SynMate. There's another seminar series I'm currently planning for young people thinking about industry job and startup. But I probably will not start until next year because my schedule is so busy, and also I also need some nomination for that seminar series. His kindness allowed another young people to give a talk during this season and next year's season. Such caring, not money, is what we really need to make this world better. To go far, go together. This is the key to diversity, equity, and inclusion. Young people and this seminar series show it by action, not by just saying. So I thank all so much, and it's my great honor to be connected with all of you, all of the world right now. Okay, it's my tremendous honor to introduce very briefly our keynote speaker, Professor Paul Weiss. He's a UC Presidential Chair and Distinguished Professor at UCLA, and also founding and current editor-in-chief at ACS Nano, meaning he will give new perspectives on a new topic to expand the synthetic biology field and make it truly interdisciplinary. Professor Weiss, the virtual floor is all yours.
Thank you so much. So I really want to thank Professor Moon for putting together this series. One of the things that we've really missed during COVID is the opportunity to visit each other's institutions and go to conferences. One of the things that I always do on those visits is ask my hosts or ask the other attendees at a meeting, who were the young rising stars in the field. And I do that both out of my own interest to see, what their ideas are, but also at ACS Nano, where we're trying to define the future of the field, we're always looking for groups that are not yet known, who have great ideas, who can share them with a community and really get people engaged in what they're doing. Every month we rate ourselves by going over the issue. We had one come out on Tuesday, and we have our meeting actually tomorrow morning at this time. And we look at what new authors we've been able to capture. And all our 30 editors, when they travel all around the world, are always looking for these young stars. And so we've struggled with what is it we can do over this last year and a half where travel has been so restricted. In the ICANX series, which I help host very early in the morning, my time, broadcast out of Beijing Friday evenings, we set up a Rising Star series for just this purpose as well for the materials community. So I'm really delighted to find this synthetic biology community mining their young stars thanks to Professor Moon. And so I'm looking forward to this series. Of course, the nanoscale is the scale of function and biology. So there's tremendous overlap in these areas. We at ACS Nano, and I personally helped put together both the Brain Initiative and the US Microbiome Initiative with this idea that nano has contributions and that we can also learn from biology and synthetic biology. Essentially, we're merging those fields. So I'm looking forward not only to today's talk, but to this entire series. Thank you so much.
Wow, thank you so much for your insightful new vision and remark. Now I clearly see synergy between nano and bio, and then there is no boundary or kind of barrier between field right now, and then this is truly interdisciplinary, and then I believe going together basically the key to solve all the problem we currently have. So thank you so much, that insightful remark and the vision, and I'm truly grateful to you join us to give a very nice kind of overview of your kind of vision and also the next thing we need to do that. Okay, so now the main speaker of today. It's my great pleasure to introduce our young speaker, Vikas. Vikas is a postdoctoral fellow from Professor Nar's lab at Tufts University. As a postdoctoral fellow, he is involved in engineering enzymes for inborn errors in metabolism. That's a very rare disease, but that's also very critical disease, I believe. And engineering yeast for non-native sugar utilization using semi-synthetic galactose regulon, and studying efficacy and application of antimicrobial peptide in mixed microbial setting. He completed his PhD in biotechnology under Professor Pale at Indian Institute of Technology, Mumbai. During his PhD, he studied the biochemistry and evolution of microorganisms involved in metabolism and xenobiotics. With that, Vikas, take it away.
Thank you. Good morning, good afternoon, and good evening, wherever you are. I'm Vikas Trivedi, a postdoc in Professor Nayar's lab. And before I begin my talk, I would like to thank Professor Moon for giving me this opportunity to present this work at such an exciting platform. I'll be discussing today some of the efforts that we have been leading in the field of enzyme engineering. Enzyme engineering, as one of the articles in Nature put it, that it has experienced various waves of technological and innovation and advancements over the course of the time that we have been studying enzymes. And whatever engineering efforts that we are experiencing in enzyme in today's world has been a product of some of the fundamental studies that were initiated way back in the early 19th century, and the early 20th century, where the enzymes are studied at a fundamental level, and the applications were designed for those basic enzymes. For instance, right, the proteases were used in development of detergents, and similarly, the penicillin acylase was developed for in synthesizing semisynthetic antibiotics. The next wave of advancement came in the early 70s and 80s when the molecular biology was developing and molecular cloning was starting to become a routine process in the labs across the globe. And between this period of early 1900s to 1970s, the structural biology was taking a hold, and the enzyme structures were being solved more often than not. And this structural insights gave us understanding about how the substrates would interact with the enzyme and which are the residues that are important for the catalytic activity. And using that as a basis, rational engineering of enzymes came into being. And one of the pioneers in that area was Professor Michaelis-Menten, who developed this approach that we know of today as site-directed mutagenesis. And using site-directed mutagenesis, enzymes could be engineered where the lipase was further engineered to synthesize a blood pressure drug, and carbonyl reductase was engineered to synthesize statins, which are responsible for cholesterol lowering and in sense has a therapeutic application. However, during the time when Professor Michaelis-Menten received his Nobel Prize in 1993, in a lab at Caltech, Professor Frances Arnold was quietly revolutionizing this field of enzyme engineering via directed evolution. She started asking difficult questions to the enzyme to try and see if she could engineer newer chemistries and newer functionalities which were never seen in biology before. Since then, and from that to the point when she received her Nobel Prize in 2018, we have experienced another wave of advancement in terms of computational approaches being applied to study the enzymes. And using this approach, I'll be talking as to our effort in engineering one of the enzymes that is involved in therapeutic application via enzyme replacement therapy. At Nair Lab, we majorly focus upon enzyme engineering development campaigns for two major disorders, galactosemia and phenylketonuria. Now, both these disorders are actually a subset of disorders broadly referred to as inborn errors of metabolism. Now, this inborn errors of metabolism arise because of the genetic mutation, which leads to the deficiency of an enzyme function. And if this mutation disrupts the enzyme function, then that enzyme cannot convert the substrate to a product. And because of that disruption, either the accumulation of the substrate or the absence of the corresponding product disrupts the bodily function, and that disruption manifests as disease. In fact, as of last week, in the IEM database, 1,615 diseases have been reported, which come under this broad category of inborn errors of metabolism. Now, in today's talk, I'll be focusing upon the efforts that we have undertaken to engineer enzyme for this specific disorder, phenylketonuria. Phenylketonuria is, like I said, is an IEM and a genetic disorder which arises because of the mutation in the enzyme phenylalanine hydroxylase, or referred to as the PAH. Now, phenylalanine hydroxylase hydroxylates phenylalanine in the presence of oxygen and biopterin to tyrosine. The PKU was first discovered in 1934, and it took nearly 2 decades to come up with a screening approach for the PKU. The incidence rate for this PKU is approximately in 1 in 10,000, but this can vary based upon the region where the incidence rate is being sampled. In fact, this disorder was so debilitating in a sense that it actually triggered a process of making the newborn screening test mandatory across the globe, wherein after the birth, within the first 2 days of birth, a blood test is performed where the blood is sampled through the heel of the baby and onto the card referred to as a Guthrie card, And that card, which contains the blood sample of the baby, is then checked for various IEMs. So because of this deficiency of this enzyme phenylalanine hydroxylase, the phenylalanine is unable to convert to tyrosine, and which leads to accumulation of phenylalanine in the body. And accumulation of phenylalanine in the body has now known to cause severe intellectual disability, to the extent that the lifestyle of the person suffering from PKU is severely compromised. So what are the treatment options and current management approaches that has been used for PKU? In the early 1960s, when this screening test was employed, whenever the newborn was diagnosed with a PKU, a strict diet control was implemented, in which case a specifically medicinally formulated mixture of amino acid was given as a supplement, and the intake of the protein was very strictly and carefully monitored to avoid intake of protein at any given day of more than 6 grams. This involves major lifestyle changes because, as we know, as a PKU patient, the person would not be able to eat anything or everything that they feel like. And because of this major lifestyle changes, it becomes difficult to strictly comply the diet, and falling out of this compliance again results into the cognitive disability. Another treatment option that is currently actually available in market is Kuvan. Now this Kuvan is in the form of a cofactor supplement, biopterin, which though effective in managing PKU is not beneficial to all the PKU patients, as it is known that only approximately 20% of the patients respond to this treatment. In fact, as of today, more than thousands of mutations have been reported in the PA gene, which can contribute to PKU. And because of that, some of the patients would not respond to this Kuvan treatment. For those patients in the market, currently an enzyme replacement therapy is available in the brand name Palynziq. Now this Palynziq employs the enzyme phenylalanine ammonia lyase, which is different than the PAH enzyme, the hydroxylase enzyme, which is natively found in the organism. And this enzyme is injected as a pegylated formulation. However, the high doses of injection of the PAL enzyme or this peg-valiase enzyme has been shown to have an adverse side effects. Moreover, because PKU spectrum ranges from mild to severe disorder, the varying dosages of this PAL enzyme is required for those patients. And in some cases, because the treatment is capped at 60 mg per day, some of the patients are removed from the treatment program because they do not respond to even such a high dose of PAL enzyme or peg-valiase enzyme, which essentially begs the question, can we engineer this enzyme to have a higher activity, which could help those outliers of the patients? And also, because of this increase in activity, we could probably lower the dosage and compensate for the adverse side effect that develops during the treatment. So this phenylalanine ammonia lyase, which is being used in the peg-valiase treatment, is derived from organism Anabaena variabilis. From here on, I will refer to that enzyme as AVPAL star, which is a double mutant variant of a parental AVPAL enzyme. Now, this phenylalanine ammonia lyase performs the non-oxidative deamination of phenylalanine to tranexamic acid. And this tranexamic acid, during this process of conversion of tranexamic acid, a free ammonium is released. Though this enzyme has been extensively studied at a structural level, the engineering efforts have not been successful yet. What do we know about this enzyme AVPAL as of now? We know that this enzyme requires tyrosine as a catalytic base and contains a prosthetic group, which is referred to as 4-methylidine imidazole 5-one, common abbreviated as MIO. And this MIO group is special in a sense that it is formed as a product of post-translational modification of the enzyme, where alanine, serine, and glycine residues at the active site form a ring compound, which is involved in the catalytic activity of this enzyme. usually this phenylalanine ammonia lyase mechanism proceeds via an adduct formation where the substrate interacts with the myo group and forms an adduct, which essentially performs a deamination of phenylalanine to form the tranexamic acid. And like I said earlier, this enzyme is less active and suffers from a low turnover rate of kcat, approximately 1 per second. So how can we engineer this enzyme, and what can we do about to improve the activity? Most of the engineering campaigns would require some kind of a screening and a selection process. One of the processes that was developed for the engineering PAL enzyme utilized a biosensor, which is a synthetic circuit that responds to the level of tranexamic acid and gives a response which can be related or linked to the improvement in the enzyme function. But so far, the reported attempt with the biosensor has not been successful and has not yielded an improved enzyme variant of PAL. So we essentially focused our efforts on a growth-based screening. And how can we implement the growth-based screening for the improving PAL activity? Like I said, PAL enzyme releases free ammonium ion during the conversion of phenylalanine to tranexamic acid. Now, this release of free ammonium can be coupled to the growth where ammonium can be assimilated in the central carbon-nitrogen cycle and a biomass can be generated. Based upon this, Zach, who was a PhD student in a lab, formulated a minimal media enrichment selection media, which involved supplementing the minimal media with phenylalanine as the sole nitrogen source. Now, when we look at the growth of the E. coli strain carrying an inactive variant of PAL, we see some amount of growth taking place, which indicates that E. coli per se has some basal ability to utilize phenylalanine as a nitrogen source. However, when we complement this E. coli with an active variant of AV-PAL, we see further risking of the growth and further improvement in the growth rate, which we exploited in our current study to select for our improved AV-PAL variants. So the whole approach can be summarized in this particular slide, and I'll walk you through as we go along the data. So the first step involved generating the library for the AV-PAL and enriching that using our growth selection method. Dr. Karishma Mohan, who was a postdoc in our lab, generated this AV-PAL library using a random PCR mutagenesis approach and performed a selective enrichment of that library in a minimal media containing phenylalanine for 3 subsequent passages. At every passage, we collected the library pool and prepped out the plasmids from those passages and performed the next generation sequencing to understand what we call it as a deep mutational landscape of the enzyme. Now, what exactly is deep mutation scanning? It is one of the very recently developing field of protein engineering, where it gives us an opportunity to perform high throughput screening of the enzyme variants across the length of protein. And because of this, we can predict to some degree of accuracy as to what would be the changes that can occur or what benefit will the enzyme experience based upon the changes in the amino acid that we are sampling. In this case of a deep mutation scanning, or commonly referred to as a DMS, fitness is used as a metric. In a simple sense, it measures what is the benefit that the variant amino acid will provide compared to the wild type at that particular position. Now, using this DMS approach, we asked a question to begin with as to what is happening now that we have a DMS landscape of this AV-PAL enzyme. What exactly is happening at the conserved residues of this enzyme? Like I said in the earlier slide, we have significant information about the structure and function of this enzyme. So we know precisely which residues are involved in the catalytic site and which residues are involved in the substrate binding and for the catalysis. When we looked at the permissivity and restrictivity of those positions based upon the amino acid that we sampled, we not surprisingly saw that most of the positions were restricted to any changes. Now, what you see over here is a heat map of this enzyme. where dark blue indicates a reduced fitness because of the mutation or changes of the amino acid, and bright red spots indicate positive fitness because of the changes in the mutation at that particular position. And like I said, not surprisingly, the Mayo position, which is the catalytic center of this enzyme, had highly restrictive behavior in terms of the changes or mutation at that particular position. Interestingly, we did find some positions where there were increasing benefit, what we were observing because of the changes in the amino acid, and which was interesting to note because that has not been reported till date for these positions for the PAL. So since DMS essentially involves understanding the whole mutational landscape of the protein, when we look at all the fitness of all the amino acid changes across the length of protein, again, not surprisingly, what we see is that most of the mutations over here represented in this fitness map have a negative fitness score, which indicates that most of the changes are detrimental to the enzyme function and do not contribute to the enzyme activity. Whereas few of those positions or few of those mutations that we are seeing over here provides positive fitness score indicating improved benefit or improved enzyme function. And we had some preliminary data from this mutational library that we had generated. And when we looked at the enzyme activity of those purified variants and looked at the correlation of those purified variants with the fitness score from this current DMS landscape, we saw a pretty decent correlation between the activity and the fitness score, indicating that in our study, the fitness score can essentially be used as a metric and a reflection of the enzyme activity for AVPAL. As you can see, there are a lot of changes that are happening which show positive fitness across the length of protein. So how could we identify the sites that can be selected for next generation of mutagenesis campaign? Now, when we map those positively fit and the negatively fit residues onto the structure of protein, which is shown over here, you can see that most of the positively fit residues indicated in red are surrounding the active site. And here you can see the phenylalanine in yellow, which is marking the catalytic center. However, interestingly, some of the positively fit residues were farther away from the active site of that subunit at a very distal end of the subunit. And why would this happen? To understand that, it might be helpful to know that this protein is an homotetrameric protein, which means that 4 subunits are required to form a functional enzyme. And when we further take a look at those subunits, we see that subunit A and subunit B are oriented in an anti-parallel fashion. The red center that you see over here is a catalytic site, and you can see that the catalytic site of both the subunits are facing away from each other, indicating that catalytic site is actually formed by multi-subunits coming together, which might explain as to why these positively fit residues are observed in our current mutagenesis campaign. Further, when it can be emphasized from the fact that when we study the distance of those fit residues from the phenylalanine, we see that most of the fit residues are closer to the phenylalanine, dot phenylalanine of the subunit, but some of the fit residues are actually closer to the other subunits of the AVPAL enzyme, which is a very interesting and an intriguing problem in itself to study. So based upon this, we identified few mutational hotspots for second generation mutagenesis campaign. And to pick those hotspots, we applied a regression onto the fitness score across the passages and picked these 7 residues as the potential hotspots for next set of engineering to find improved variants. And to achieve that, we implemented multiple strategies for that. One of the approach was to actually generate site saturation mutagenesis library which samples 20 amino acids for each and every position individually and then perform a DMS onto that. Now, if you recollect in the initial heat map that I had shown, there were a lot of gray regions, and those gray regions were the amino acids that were not sampled in our error-prone PCR campaign. because error-prone PCR is known to sample only 6 amino acids out of the available 20. So we wanted to see as to what is the fitness of those other non-sampled amino acids. And to understand that, we performed the site saturation mutagenesis approach for those 7 positions. And what you can see over here is a heat map for those 7 positions for all the 20 amino acids. And like in the other plots, the dark blue indicates least fit positions or least fit changes, and the bright red indicates most fit changes. You can see that for the most of the 7 mutational hotspots, the spanning of amino acid is pretty restrictive. However, T102 and N453 shows decent permissive behavior. So, and this, because this enzyme is very well studied in literature, there are a lot of homologs available in the literature. So how does this permissivity behavior compare with respect to the natural diversity for the EV-PAL homologs? For that, we generated a consensus for more than 1,000 homologs of EV-PAL and looked at the permissivity of mutations across those homologs. Now here what you see is a tiling of those 7 positions where we are looking at the natural diversity, and which has been graded from dark green indicating most frequent to light green indicating least frequent. On the top panel, what you see here is the amino acid that we have sampled in a current directed evolution campaign. And the residues highlighted in yellow are indicating that they are observed in the natural homologs as well. As you can see, we are definitely sampling newer amino acids at those positions which are otherwise not observed in the natural diversity, which is very interesting to note. Now that we understood the effect of those 20 amino acids at those 7 hotspots, we wanted to see if we can generate a combinatorial library for these 7 positions and identify an improved variant which can serve for the ERT application. Now, for that purpose, we employed 2 additional strategies which involve combining at any given, for any given variant, 3 mutations out of those 7 positions, which essentially indicates 7 choose 3 library approach. So any variant will have mutations spanning from 1, 2, or 3, but not more than 3. And in the other strategy, we employed 7 choose 7, where we attempted to mutate all the 7 positions in the protein at for a given variant. So in this sense, this library would contain mutations spanning from 1 to all the way up to 7. And we know that in some sense, multiple mutations can be detrimental to the enzyme function. So we wanted to cover our bases by spanning range of mutations ranging from 1, 3, and 7. Using this 2 strategy, we could essentially identify improved variants of EV-PAL. Now here I'm showing you some of the kinetic characterization of some of the variants that we had selected using those 2 strategies that I described earlier. Now this is the Michaelis-Menten plot for those variants. The one that you see in black is the parental EV-PAL type of parental EV-PAL enzyme, and which has a catalytic activity of 1 units per mg. Whereas there are some of the variants, T102E referred to here in green, has a 2.4-fold improvement over the parental EV-PAL enzyme. And another variant T102R M222L D306G has approximately 2.3-fold improvement compared to the parental EV-PAL enzyme. This variant, green variant, I'll be referring more commonly from here on as a double mutant because it contains 2 mutants. And the mutation in pink, which contains 3 mutations, I'll be referring as a triple mutant from here on. Other thing that would like to point out here is that because we are potentially looking for an application of these enzymes for enzyme replacement therapy, one of the problems that has been observed for these enzymes is that they exhibit product inhibition, which means that tranexamic acid that gets formed in the reaction can inhibit the reaction and reduce the conversion of phenylalanine to tranexamic acid, which can be problematic for the therapeutic application. For that purpose, we studied the inhibition kinetics for the top 2 variants, the double and the triple mutant, and compared their profile with the parental type. where we studied the rate of reaction across different concentration of tranexamic acid and looked at the inhibition pattern for those. As you can see over here is that the parental AV-PAL enzyme exhibits IC50 for tranexamic acid of approximately 10 micromolar, but the other two improved variants had 3 to 4-fold increased IC50, indicating lower sensitivity to or lower inhibition to tranexamic acid for the PAL enzyme, which was very interesting to note, because that would indicate that these enzymes are not only have a higher catalytic activity, but they also have a lower subproduct inhibition. So once we had identified those improved variants, we wanted to understand how these mutations are actually contributing to the improvement in the enzyme function. For that purpose, we collaborated with KCAD Enzymatic Private Limited, one of the companies back in India, who performed the computational approaches that I'll be describing in our study. It might be known that molecular dynamics is one of the most commonly used approaches to understand the impact of mutation on the enzyme function. And before we go on to understand the molecular dynamics simulations, we would like to understand how this reaction is proceeding. And if you recollect, in the earlier slide, I had indicated tyrosine as the catalytic base that is very essential for the enzyme activity of this enzyme. And along with tyrosine 314, there is this MYO group, which is involved in the catalytic cycle. And there are other residues which are involved in the hydrogen bonding network and perform the catalysis to form tranexamic acid as a product. And during this process, a transition state is achieved. Actually, multiple transition states are achieved, but we focus our efforts on the step 1, which we are referring from this point of enzyme docking the substrate onto its active site to the formation of the first transition state, which is referred over here. So, and you can see that because of this catalytic mechanism that we see here, tyrosine 314 and MYO are very important residues. And if we study what is happening in all those mutants with respect to the tyrosine 314 and MYO, we might gain some insight on the activity for AV-PAL and the mechanism by which the mutants are improving the enzyme function. So as I said, in this particular molecular dynamics simulation, we measured the distance of phenylalanine, which has been docked onto this PAL structure, and measured the distance of that substrate to the MYO and the tyrosine 314 for all the mutants. And what we see over here is a scatter plot for those molecular dynamic simulation. And we are indicating those scatter plots for all the mutations that we had studied we identified in the current study, and additional mutation L108G. Now this L108G has been reported in the literature to lower the enzyme activity, which means this mutation lowers the fitness and reduces the enzyme function by approximately 80%. So when we look at the distance of this phenylalanine to the MYO on the y-axis and distance of phenylalanine to tyrosine 314 on the x-axis, we would expect that in an ideal scenario, most of the scatter plots will cluster at the lower left quadrant. And as you can see for this less active mutant L108G, most of the scatters are farther away from the left quadrant, lower left quadrant, indicating that substrate is moving away from tyrosine 314 and MYO at the active site in L108G variant. Whereas in the other improved variants, M222L, 218S, and double and triple mutant, we see most of the clustering occurring in the lower left quadrant, indicating that in these mutants, the substrate is moving closer to the MYO and 314, which would favor an improved attack conformation for those improved variants. So with this molecular dynamic simulation, we gained some insight as to how these mutations are improving potentially the enzyme activity. But as any enzymologist would know, that enzymes catalyze the function by lowering the transition state energy barrier, which essentially is the roadblock in forming substrate to a product. And we wanted to study what's happening in this transition state 1 for all those mutants and see if we see a pattern emerging in the transition state energetic barrier. For that purpose, we performed something referred to as a quantum mechanics and molecular mechanics simulation, commonly referred as a QMM analysis. And using the QMM analysis, we estimated the energetics of the enzyme reaction starting from binding of the enzyme to the binding of the substrate to the enzyme and up to the formation of transition state. Now, when we look at the energetic landscape, over here, what you can see is the y-axis indicates the free energy of the various steps of the formation of the transition state 1 and the IS1 state, which is indicated here. And you can see there's a barrier of 13.4 kilocals per mole for the wild type AVPAL enzyme. Now, when we look at the L108G variant, which is the less active variant, not surprisingly, and actually interestingly, we actually see an increase in the transition state barrier for that particular variant. Now, so we also wanted to see what's happening for the mutants. And you can see that for the mutants that we identified in the current study to have a higher activity, they exhibited lower transition state energy barrier, which meant that these mutations somehow stabilize the transition state in a much more favorable manner and made the reaction proceed to the formation of tranexamic acid in a much easier fashion compared to the parental type. So what happens if we try and see a correlation between this transition state energy barrier and the enzyme activity, which is what I'm indicating here. You can see that the y-axis indicates the catalytic activity of the enzyme as we determined from our experimental study, and the delta G, which is the transition state energy barrier, which we determined from the computational study. The L108G, which is the less active variant, is right at the bottom over here, has the highest barrier and the lowest activity. What we can see is that some of the other variant, the parental isotype or parental AVPAL variant and the higher active M222L variant exhibits decent correlation, where we see that the drop in the transition state energy barrier is correlated with the increase in the activity for those mutants. However, for the other higher active variants, what we observe is, though the transition state energy barrier is reduced, the activity does not change, which potentially indicates to us that in these mutants, now the bottleneck, which was initially at the transition state 1, has moved to the other downstream steps of the enzyme catalysis, which is one of the aspects of study that we are undertaking in the coming time. So in this particular data, you can also see that N453S, which we were expecting to have a higher activity, does not show an improvement in the KCAT at all. So what's happening in the N453S there? This residue, N453S, when we map it onto the structure, we see that it is close to the surface of the active site. And because it is close to the surface of the active site, we studied the role of that particular mutation in tunneling the substrate from the surface of the active site to the catalytic site of the enzyme. And we compared that energetics of substrate tunneling with the parental type, and we attempted to see if there's any difference. What you can see from this plot is the PMF force of pulling of substrate from the surface of the enzyme to the catalytic site, and more negative the value, better is the pulling and better is the binding energy of that enzyme for the substrate. And you can see over here that N453S exhibits improved binding energy and has an improvement in the binding energy by 35 kilocals per mole when compared to the parental type. So if this is really binding better, can it improve the diffusion of this substrate into the enzyme active site? And if yes, what is the improvement? To understand that, we generated different variants of AVPAL and combined those mutations with the N453S. What you see over here is a kinetic Michaelis-Menten plot for the parental type and its N453S mutant. And you can clearly see that there is barely any improvement in the, there is no improvement in the active site when N453S is added to the parental type. We also attempted to study the effect of that mutation onto the other higher active mutants, double mutant and the triple mutant. What you can see is that in the double mutant, adding additional mutation of N453S, in fact, brings down the activity back to the parental type. Whereas in case of a triple mutant, the adding of 453S completely abortions the catalytic activity, which was surprising to us because we observed a positive fitness for that particular mutation. Now, why would this be happening? Right? So because our fitness of calculation was derived from the whole cell enrichment of the library, we have posited that maybe this mutation is providing some benefit in the whole cell manner, not at the pure enzyme level, but when it is present inside the cell. So when we looked at those variants, same variant that we verified earlier at the whole cell level, what we see over here is the TCA production for all those variants. And we compared the TCA production for those variants to the parental types listed over here. What you can see is that when we add N453S to the parental type, we see a significant improvement in the TCA formation at the whole cell level. And this improvement is observed even for M222L and the double mutant. Interestingly, G218S and the triple mutant had complete loss of activity when the N453S mutation was combined. However, there's still improvement at the whole cell level can explain as to why we observe positive fitness for N453S during our mutation and enrichment campaign. And because addition of N453S further ups the performance of this double mutant, it can serve as a potential candidate for probiotic therapy, which involves injecting or giving the PKU patients with the whole cell containing the enzyme. So to conclude our work, in the current study, we identified 79 functionally relevant sites for AVPAL engineering, which is one of the most thorough and investigation that has been undertaken till date. From those 79 mutation functionally relevant sites, we studied 7 hotspots in detail and identified 2 variants with improved KCAT, which has the potential to use in enzyme replacement therapy. Using the computational approaches, we gained some understanding as to how those mutations are improving the enzyme activity. By now, what we know is that these mutations generates an improved attack conformation and lower the transition state barrier. And among those mutations, when we add 453S, the double mutant increases the performance of the whole cell by 6-fold compared to the parental type, and which means to us that this can be a potential candidate for probiotic therapy. And these variants have commercialization potential in the therapeutic market. And based upon our current study, we have some interesting development where we are spinning off a startup based upon these variants, which we have aptly named as an Enrich Bio. And this comprises of the team, Todd Chappell, who was a postdoc in our lab, who's the main lead and a co-founder of this company, and Professor Nair and me are serving as of now as a scientific advisor for this Enrich Bio. Lastly, I would like to acknowledge Professor Nair for giving me an opportunity to work in his lab and being very patient with me during the course of my time. Dr. Todd Chappell, who is a postdoc and a co-author of this present study, helped immensely in all the effort that we have taken for studying this enzyme. Zach and Karishma actually laid the foundation of the work for this PAL engineering efforts. And I would also like to thank all the other lab mates of Nair Lab. Atreya, Beck, and Reina are also involved in engineering PAL currently in our lab. We would also like to thank Tufts University and NIH for the funding, Professor Moon for giving me this opportunity. And with this, I would like to take any questions.
Wow. Fantastic talk and work and amazing. I mean, thanks so much. So personally, very interesting, topic to me, of course, because my lab also developed very specific sensors for phenylalanine to detect PKU. And we are doing also many computation prediction-based protein engineering work. now to improve the enzymes and the transcription factors. Okay, so I found some question, one of the genius colleague, Chang Lu, and then I guess this is a very tough question, I believe. So he said, great work, how much epistasis is revealed in your mutant combinations from the DMS single mutants? More generally, do you think DMS on single mutants, which captures no or little epistasis, is enough to give the key mutations worth combining? And there's another question. So could you answer that question first?
Yes. Yes, so I totally agree with Professor Changlu here that single mutants cannot really inform our selection of combining those mutations. And that is the whole reason we went on to employ multiple strategies where we were combining 3 mutations or 7 mutations in a given variant and using all the 20 amino acids at that particular position. We just use those single mutations as an indicator, but not as a complete basis for engineering our enzyme. And to answer the epistasis part of it, because we had used Illumina sequencing platform for our current study, we obtained short reads. And because of the short reads, the physical linkage of this mutation, the information of the physical linkage of this mutation is in some sense lost. But in a preliminary understanding or investigation of the epistasis, what we observed is that some of the beneficial mutations, when they come together, they are detrimental for the activity. And some of the mutations which are beneficial in isolation, they have additive effect when they come together, which is again not surprising for an enzyme engineering campaign.
Okay, wonderful. So another question from Chang Lu again. If you don't mind, what is the happening in whole cell that is not captured with your enzyme kinetics assay? Is it expression or the rapid use of the liberated ammonia?
It could be, so we haven't really thoroughly investigated that particular problem as of now, but our current hypothesis is that because TCA that is formed in the reaction can not only inhibit the enzyme, can also inhibit the product, inhibit the E. coli for its growth. And in addition, the concentration of phenylalanine that E. coli experiences inside the cell is very different than what we use to measure the enzyme kinetics. And what is known so far is that E. coli, intracellularly, the E. coli concentration for phenylalanine is approximately in the range of 10 to 100 micromolar. And it is possible that this variant performs better at that intracellular concentration of phenylalanine than those other variants, and might explain the effect that we are seeing at the whole cell level. Also, there's a possibility, like you mentioned, that the expression might have changed for that particular variant.
I see. Thank you. So also I found some question from chat, actually, not Q&A box. So this is from, I believe, Damayat. And the question is, thanks very much for your cool talk. Instead of evolving single enzymes, is it possible to evolve enzymes in a synthesis pathway? I guess he also kind of thinking about degradation pathway, so as to optimize their activities to overcome metabolic pathway bottlenecks rather than optimizing transcriptional expression. I guess this kind of question kind of a little bit broader kind of question, not necessarily to your own work, I guess. Can you?
This is beyond my expertise. I'm sorry.
Sure. So can you give the answer for that question?
Yeah, this is beyond my expertise in some sense to answer this question. So I would not be the right person to answer this.
Okay. So if not, anyone also have any other question of the Professor Weiss? Do you have any question or feedback, anything?
I thought that was a terrific and exciting talk. We've worked on the sensing side in phenylalanine, and actually I was going to reach out to you after the seminar, so as not to take our audience up with that. But this is such an interesting and I think under appreciated area
Sure.
with tremendous opportunities. So I'm really glad to see you pursuing both the science and the, the possible translation of this work.
Sure. So that's
nice.
wonderful. So if we have 1 minute, actually, I have question. Basically, as you know, Synology, based on, of course, Cambridge near Tufts University, successfully finished the clinical trial phase 2 recently by using PKU treating probiotics. So what would be the remaining challenges? I mean, they also kind of show a great promise in this area to move your work to the next level, potentially for real practical application. I believe, you said, I mean, you currently have the company right now, but what would be kind of current challenging to really kind of for the practical application? I'm super excited. I mean, you have wonderful enzyme right now, but to think about, beyond, I mean, you kind of have multiple challenges, I mean, right now.
True. So to begin with, Synlogics is working with another enzyme called as derived from another organism, Photorhabdus luminescens,
right?
Which commonly called as a PL-PAL.
Mm-hmm.
And they have tried to engineer that enzyme using, again, a sensor-based approach. And they have found like a 2-fold improvement at the whole cell performance. And like you can see in a current engineering campaign, we have a variant which exhibits 6-fold improvement over the parental type. So definitely there is an interesting development at that end. But our study is limited right now to Anabaena variabilis PAL. Mm-hmm. And some of the effort that right now we are undertaking is to see if we can translate those information or understand that we have gained from this study to the other homologs which are currently in market or can be in market for the treatment of PKU. But that study, that is one of the our study that we are taking in the lab as of now.
Okay, that's
so
fantastic.
One of the challenges that we might experience is that those mutations may not really translate to the other enzyme, but who knows? It's
sure. I mean that your work could be helpful for other people. I mean, working on the same enzyme or some other enzyme as well. So that's fantastic. So any other question from audience or Professor Weiss? Any other thing? Anything you wanted to kind of add more, Professor Weiss?
No, I was just going to say thank you again for beautiful talk and good luck in your search for a faculty position.
Right. That's the whole point of this seminar series. I mean, you will be busy, I guess. Also, hopefully you get lots of interview, and because this is important work, so hopefully you get lots of interview offer, and then finally kind of getting a job here or anywhere you want it to go. And good luck. And then I believe we have the tradition, or the professor actually already tenure from this SynBYSS alum, and then I hope hopefully your career kind of bright down the road. Okay, if you don't have any other question,
thank you so much.
I wanted to kind of close. So next week, of course, I mean, we will have Professor Nathan Price, CEO of Longevity. I mean, he started kind of some company recently. And then also Professor Navin Polkatra from New York Institute of Technology. And then I believe he's also pretty new for that job. So see you all next week at the same day, time, and as today, we'll have a follow-up discussion shortly with the Zoom link shown in the chat box. I guess, I mean, that's all the way up. I kind of posted in the chat box, and then, so see you all today or the next week. And thank you so much for attending.
Thank you.
Thank you. So Professor Weiss, I mean, you kind of asked some question directly to me, but we are actually, what my paper is under review, and then we develop. So my paper work is basically we developed the sensor that very specific to phenylalanine, but we use the biological system. And then I believe you kind of sent some link.
Yeah, I sent you a paper from ACS Sensors where we used aptamer functionalized field effect transistors to measure phenylalanine.
So our
so
sensor is
yes.
purely the biological, but that's very, very specific. And then even we differentiate between phenylalanine and tyrosine. That the
Oh, yeah,
only
no, say, yeah, the aptamers also do that. We always check the precursors and metabolites. So they were developed originally for chemical neurotransmitters in Professor Ann Andrews' lab.
Exactly. So
as part of the BRAIN initiative, but then we showed with our collaborator at Columbia, Milan Stojanovic.
that's wonderful.
Specific phenylalanine measurements.
That's wonderful. So our current approach is because we are using probiotic bacteria, we could potentially use this bacteria, the probiotic bacteria therapy, and that one detect the phenylalanine. In that way, we kind of respond to phenylalanine problem in the gut. And also the other application is we are currently collaborating with the people kind of working on wearable sensor. So potentially we kind of wanted to kind of go in that direction. So paper currently under review, and then the last revision already done, and then we are have very nice kind of review, and then minor revision right now currently under review right now. So hopefully soon the paper out. I mean,
very good.
Yeah.
Well, you can once you turn off recording, you can tell me where it is.
Sure. Yeah.
I don't want
okay, but once we stop recording,
you should. Yeah.
Once we stop recording, I'll type the chat, because once we stop and then
No, that's what I mean. We can delay that conversation.
Yeah, so I can kind of put the journal name right now to you. So currently under review in this journal.
Well, I will look forward to it when it's out.
Yeah. Actually, unfortunately, my paper is rejected from a little bit better journal. I mean, but finally, I love that journal. I mean, I love that journal, and then we are almost done, basically.
Great.
Okay. Any other thing? So if not, thank you so much again. I mean, this is great. I mean, you kind of stay until even beyond the 1 hour, and then giving a comment, and that's kind of the most valuable thing, for young people wanted to hear. And then I believe, I mean, when you become professor, thinking about ACS Nano, because now my colleague actually submitting paper to ACS Nano, some material production using biosystems. So
yeah. Shelley Claridge had a big paper with us a couple of days ago.
Exactly.
So she was one of our rising stars, and your colleague in chemistry and biomedical engineering.
So you now have connection with him, in person right now.
Exactly.
So submit the paper to him, and then I may have some paper to submit to ACS Nano. Why not? And then wearable and some other thing, and then I have some collaboration with the people.
Yeah, one of our editors is Ali Javi, who's very active in that area, and also Ildu Kim at KAIST works a lot.
Exactly. Right. Okay, thank you so much. I believe I also need to entertain some people probably waiting for me to another Zoom link. So because please join the another Zoom, and then Professor has another commitment. So thank you so much. I mean, we'll see. I mean, somewhere later, in some other real physical location rather than Zoom, I guess.
Okay, terrific. Thank you so much.
Thank you so much. I mean, have a nice day.
Thank you so much.
So the Zoom link is the top. So click that one, and then you could transfer to that one. And then basically Tony will close once we click that one.
Okay.